Hello students and welcome to my second video on Unit 1, the Chemistry of Life. This video will cover topics 1.3 to 1.6, which deals with biological macromolecules. That would be carbs, lipids, proteins, and nucleic acids, right? And they form the basis for life, which is why it's called the Chemistry of Life. Just like all my videos, this is not for profit and is just meant to help students. If you're looking for water and hydrogen bonding and elements of life, please see the video before this one. Here we go. I look at my learning guide and it says that we need to describe the properties of the monomers and the types of bonds that connect the monomers and biological macromolecules. So if I do this, I can think about monomers being like an individual Lego piece. And then I could think about polymers being that completed Lego truck. Here's another good picture of it. So those monomers are the building blocks and they make the polymers. The building blocks of carbohydrates are saccharides. The building blocks of proteins are amino acids. The building blocks of lipids are fatty acids. Some people say lipids don't really have a monomer. That's fine as well. It's fatty acids attached to a glycerol molecule. And then the building block of nucleic acids are nucleotides. Let's check out this picture here. Can you describe dehydration, synthesis, and hydrolysis? So here, use your words. Synthesis means to join together, right? So we're joining together these monomers, these blocks down here, into this nice chain up here. When that happens, it dehydrates. It loses a water molecule. So dehydration synthesis is joining two monomers together into a polymer, and a water molecule is removed in the process. Hydrolysis, the word lysis means to burst or to break, and hydro means with water. So it means adding a water molecule with the help of an enzyme to break a covalent bond between two monomers. So in other words, it would be to break apart this long line here into their constituent monomers. So if we went back to the Lego truck, right, if my son wanted to break this down and make something else out of it, that would be hydrolysis. Putting it together would be dehydration synthesis. Breaking it down would be inserting a water molecule. Putting it together is removing a water molecule. Let's see it in action here. Here is dehydration synthesis for two glucose molecules. And so these are monosaccharides. So here I have a model of it. Hold on one second. Thank you. Now I've got my model. So just like here on the screen, you can see I've got an O and an H. And here is a carbon with an OH as well. So I would encourage you to do something like this. We'll do it in class if you're a TASM student. Here is the H and the OH. They're going to get pulled off. So they get pulled off. That is a water molecule. And now these are going to be bonded on that oxygen. This is a covalent bond. This is very strong. So notice how the two monomers were put together with the help of an enzyme. And this OH and this H were pulled off and they are bonded on the O. If you think back to the previous video about electrons, this will fulfill the octet rule for that oxygen because now there will be two bonds that are shared, a covalent bond, and it had 6 electrons in that outer shell anyway. This forms maltose, which is a disaccharide sugar and a water molecule. So that's dehydration synthesis. Here, hydrolysis with the help of an enzyme will cleave this bond. It's going to break a covalent bond. Remember, that's a strong bond. It adds in the water molecules, and it adds the OH to this one and the H to this one. You don't need to worry about where does the OH or the H go. You can just see that that this O has now fulfilled its bonding requirements, and this O has fulfilled this one as well. All right, and so that's hydrolysis, breaking down a polymer into two monomers, requiring a water molecule. All right, that wasn't too bad for 1.3. Now I look at 1.4 and I see, okay, it's still talking about monomers and the bonds that connect them. So we're going to give them some names that we need to know. And the reason we give them names is that way we know which macromolecules we're dealing with. But then if I look at the essential knowledge in the unit guide, and I really encourage you to read these unit guides and see if you can understand what's going on in them, we're seeing these words structure and function. And that is super important in biochemistry and chemistry in general. And you can think that the structure of a molecule leads to its function. So So that way we don't lose the big picture. Think about what is the what are carbohydrates used for? What are lipids or fats used for? What are proteins used for? What is nucleic acids used for? And so then you can think, all right, that's the function of them. How does that structure of that molecule lead to it? So here are the polymers and the monomers, just a review of carbohydrates. The monomers would be monosaccharides, prefix mono meaning one sugar, and then the polymers are polysaccharides or many sugars. If it's two sugars together, it's a disaccharide, and then if it's more than that, it's a polysaccharide. Here, lipids, once again, some people say there isn't a monomer, that's fine, but basically it's three fatty acids attached to a glycerol molecule or two fatty acids attached to a glycerol molecule plus um, the phosphate part for a phospholipid. Proteins, once again, amino acids make proteins, and nucleotides make up nucleic acids. Let's jump in. All right, here are carbs. Carbs are delicious, right? We love eating it. Bread and pasta, etc. And they are simple sugars, and they are used for quick energy in our body, right? And so, if I love running, and so if I go running for a long time and I run out of carbohydrates, I might hit the wall. Also, if I run out of carbohydrates, I start oxidizing fats, which is a good thing to keep me in shape. All right, let's get back to the real deal here. Sugars are usually in a 1 to 2 to 1 ratio. So here you can see in glucose, it's 6 carbons, 12 hydrogens, 6 oxygens. When you start building together the polymers, right, you're going to lose a water molecule each time. So here, C6H12O6, well, if we combine the two glucoses together, remember on the previous slides, we lost a water molecule. So that'd be C12H22 because water is H2O. So we lost two H's and one O. So instead of O12, it'd be O11. So if you think like if we put 6 glucoses together, it'd be C30H50O25, right? Because we lost 5 water molecules. So it won't be exactly a 1 to 2 to 1 ratio, but it will have relatively more oxygens than, say, a lipid does. Carbohydrates are made through photosynthesis, which will be what we get into with Unit 3. They often end in the suffix OSE, such as maltose, fructose, sucrose, glucose, and they're made of carbon, hydrogen, and oxygen, as stated earlier. All right, a couple more quick notes. Monosaccharides are simple sugars, or one sugar, glucose and fructose. Polymers, disaccharides are two monosaccharides joined by dehydration synthesis. Now I'm going to give you a new word. That word is a glycosidic linkage. Glycosidic linkage is when it's bonded on that oxygen, and that's just the name we give it. That's a covalent bond. Polysaccharides are many linked together, can be hundreds to thousands. These, the way that carbohydrates work is that there's different monomers, right? And as they get put together, they might have different functions. You can think about that as like if I put together one football team with all forwards, they would have a different function than if I put together a team that was all goalies. And so here I might have storage sugar molecules like starch and glycogen. And then I might have structural ones like cell walls or things that are hard to break or like the bark of a tree. All right, here's a good example of that structure leading to the function. And so glucose exists in a ring form in water. And so our body is an aqueous environment. So the glucose is in this ring form. Glucose is able to quickly enter the cellular respiration pathway to be made into ATP, right? So we need glucose in order to get energy. So if you eat some sugars, you ate a donut before watching this video, your body's breaking down the sucrose into glucose. So that way that glucose can enter your cells and help give you energy. Glucose can have two different forms. It can have the OH on this side, away from the CH2OH, or it can have the OH up here, closer towards the CH2OH. It's called alpha glucose and beta glucose. Well, if we put together a bunch of beta glucoses, we get cellulose. If we put together alpha glucoses, we get different items. And so that's just a good example of how structure leads to function. Sorry. So I just mentioned the alpha ones. If you put those together, it can make something like starch. All right. You don't need to memorize any of that. I would just say the important thing to note is that structure leads to function. Okay, we just did carbohydrates. Let's do fats. And so in this instance, you're going to have this fatty acid chain right here. It's called a fatty acid chain. It's got these carbons surrounded by hydrogens, and this is going to be nonpolar. These bonds are sharing the electrons equally. It makes a nonpolar tail here. Just like earlier, we're going to remove an OH and an H. If you remove an OH and an H, that is a water molecule. That's H2O. So that's dehydration synthesis. This is called an ester linkage. So you remove the OH and you remove the H, and then you're able to bond these two together. And this makes a fat molecule. If there is no double bonds, then that is called a saturated fat. And at room temperature, that will be a solid, such as butter. If there is a double bond, which will come up on the next slide, then that is an unsaturated fat, and that will be like oil. Notice how, just like in carbohydrates, this is also bonding on an oxygen. So we give it the name ester linkage there, so we know we're talking about fats. Here's going to be a nice little graphic of it. So here is our fatty acid. Here comes the enzyme that's going to remove the water. It's going to bond between that C and that O, and it's an ester bond. It's very strong. It's covalent. Here's hydrolysis, breaking it, inserting the H and the OH, the water molecule, and breaking it into fatty acids and glycerols. So why would we break fats? Let's say that we're out of carbohydrates and we need to burn some fats in the cellular respiration pathway to make energy for ourselves. Okay, so now there's another type of fats called phospholipids that are really, really important, and they make up the cell membrane. Phospholipids have two fatty acid tails, and then they have a hydrophilic head. And so here we go. The glycerol undergoes a dehydration synthesis with two fatty acids. So it's going to, spit out two water molecules and attach these. And then the third part of the glycerol bonds with a phosphate and other polar molecules. This will make the head like water, hydrophilic. It likes water, and the tail fear water or not want to be there. This forms natural bilayers, and this requires no energy. And so here, this is going to have the heads are going to face the outside water, and these heads are going to face the inside water in a cell. The tails will naturally arrange themselves like this as they fear the water, and this is no energy required. This is really important because it gives the cell a compartment. It's like it'd be hard for me to teach if I didn't have a room to teach in. And so it also provides for compartmentalization, right? You take your math class in your math room and science here, and unless we're trying to do something interdisciplinary, but it lets us have our own spaces. And so it lets the mitochondria have its own space, the chloroplast, the endoplasmic reticulum, and other organelles. All right. There's another category of lipids called steroids. And so these can be hormones. They can be cholesterol, which helps to maintain fluidity in the cell membranes. They count as lipids because they're hydrophobic. They don't dissolve in water, but they do have a different structure. So they're not that classic glycerol with three fatty acids. More on this to come later on. Okay, we've done carbs and we've done lipids. When you're reviewing this video or when you're thinking about it or answering the questions, think about how does the structure of a carbohydrate lead to its function? That's to be quick energy. The function could also be to store energy. We'll get into that more later on. And it could be structural. How does the structure of a lipid lead to its function to also store energy or to serve as a phospholipid, to serve as a cell membrane? All right. Proteins. When you're looking at me, you're seeing all proteins. Proteins do everything. They're amino acids. They're usually folded into shapes. They serve as enzymes. There's thousands of enzymes in a cell. They help build muscles and bones. They work to fight infections. They serve as antibodies. They fight COVID. They do everything. They're made of carbon, hydrogen, and oxygen, just like carbohydrates and lipids, with the addition of nitrogen and sometimes sulfur. All right, let's dig into it here. Here would be the monomer. The monomer is going to be an amino acid. Here you have an amino group, you have a central carbon, attached to it is a hydrogen, and then you have this side chain. And this side chain differs for the 20 amino acids. And then on this end, you have a carboxyl group. We say that proteins have directionality. They're always going to start with an amino group, and then they will end with a carboxyl group. And it's on the carboxyl group that the dehydration synthesis will happen. All right, and that's called a peptide bond. So let's go ahead and let's see some of this in action. Here's just the different side chains or the different R groups. You do not need to memorize any of these. They will give you context about what the R group is like on the AP exam. This is not. This would be an instance where memorizing it just doesn't help you. All right, so here I told you that the peptide bond is going to happen on a carboxyl group. So here's the amino group. Let's pretend that this is the start of this protein. Maybe this protein is ghrelin, right, which is a hunger protein. It's telling me I'm hungry. Here's the amino group. Here's my central carbon. Here's my hydrogen. Here's the R group. All right, it's just flipped from the last picture. This would be, if it's the start of a protein, this would be methionine. Here's my carboxyl group, and here's the OH, and here's the H on the next amino group. So it's going to go carboxyl to amino group. They're going to combine together in dehydration synthesis. So they dehydrate, they lose a water molecule, and they're now bonded between this carbon and this nitrogen. That is a covalent bond. They're sharing electrons. It fulfills the octet rule for the carbon and for the nitrogen. Nitrogen needed 3 more electrons to fulfill its valence shell, and everything works. All right. These peptide bonds will always form between a carboxyl and amino group, and there can be thousands of amino acids bonded together to make a protein. Here is another example of it. So there's the peptide bond. This is a good picture because it's showing you, look, here are those different side chains. And so we always start with the amino end as a protein. They have directionality, and they will end with a carboxyl end. As proteins are being synthesized at the ribosome, they will start to form shapes. And the first order is called primary order, which is just the order of the amino acids. It'd be like if you took the Legos out and laid them out, it'd just be the order of them. These are bonded together covalently. You're not going to break them. In secondary structure, due to hydrogen bonding, the protein will start to form beta-pleated sheets or alpha helices. Linus Pauling figured this out while he was sick one day, just like I think eating chicken soup or something and drawing it out. It's pretty cool. Remember that a hydrogen bond is between a positive hydrogen, because that hydrogen is bonded to a more electronegative element. Here is an H that's bonded to an N. The N, or the nitrogen, has more protons, and it will hog the electrons more often, making this H positive. This positive H is attracted to negative elements. And so in this instance, it's attracted to this negative O here. This positive H is attracted to this negative O. This positive H is attracted to this negative O. Over here, this positive H is attracted to this negative O. And this forms beta-pleated sheets and alpha helices. So starting to give a shape to the protein. And the most important thing is that a protein's shape determines its function or what it can do. In tertiary structure, the R groups start interacting. And so the R groups were the part that is different for each amino acid that hangs down, right? And so, back here, here's the R group. Here was a picture of the 20 different R groups. Well, R groups that are polar will like water, and they'll go towards the outside of the protein. R groups that are nonpolar don't like water, and they will go towards the inside of the protein because our body is a watery environment, and so the proteins are made in water. R groups that have sulfur in them, like cysteine, will form bonds with other cysteines. They will form disulfide bridges. Some R groups may be able to give up will form ionic bonds because they will be charged, and so then they will bond together, and there can be hydrogen bonding between them as well. All these bonds help to make the shape of the protein, and so it's pretty cool. There's a program called Fold It that you can use to play and work on tertiary structure, but it's the interaction of the R groups. This is why if there's a mutation or a change in an amino acid in a protein, then it can change its shape, right? Because it might have a different R group. We'll go over that more in Unit 6. Quaternary structure is when two proteins come together. Like hemoglobin is four proteins put together so that they can work together. And so let's go through the layers again. Primary structure is the order of the amino acids. Secondary structure would be they start forming alpha helices and beta pleated sheets. The tertiary structure is that the R groups start interacting and it forms the shape of the protein. And then those shapes lead to their functions. If we put together more than one protein or more than one chain of amino acids, then we get quaternary structure. All right. These structures can be denatured or broken down by changes in temperature and pH. These bonds are held together, and so if you change the temperature, you may affect the bonds. If you make it too hot, it will denature the protein. It's like frying an egg. If you change the pH, you change the interactions between some of the acidic and basic R groups. And so this would also change the protein. Proteins function best or have the highest activity rate as enzymes in a specific pH and in a specific temperature. For us, it's 37 degrees Celsius for our body temperature. And then depending on where the protein is, like pepsin's in your stomach, then it works best at an acidic pH. A protein that's in your blood works best at a neutral pH. A protein in your intestine, such as trypsin, works best at a basic pH. All right, structure and function of biological macromolecules. And so here, this is 1.5. And so this is dealing with how does a change in the subunits of a polymer lead to changes in the structure or function of macromolecule. So this is going to be pretty quick, right? And so here I was talking a little bit earlier about starch versus cellulose. So starch is something you can eat, that you can break down. You might like French fries. I love them. They're my weakness. Cellulose is like tree bark. You're not going to do that. If you look at them, I see the difference being OH here, OH here, OH here. The OH is always on the bottom in this one. Here, it flips. OH is here, OH is here. OH is here, OH would be up here. The difference in the monomers led to the difference in the function of the polymers. Here's a good example of it again, starch, glycogen, and cellulose. The differences in those H lead to glycogen and starch having these branch-shaped structure, whereas cellulose forms these long, rigid chains that can be stacked upon each other like tree bark. Here's an example I made on a website dealing with hydrophobic and hydrophilic amino acids. And so here would be a bunch of different amino acids put together in a protein. And so this would be the primary shape. Maybe it's starting to make alpha helices and beta pleated sheets. And then boom, when we put them all together, notice how the hydrophobic ones go towards the middle and the hydrophilic ones go towards the outside. Cysteines forming the bridges between itself. Now let's change the monomers. So this is a different protein, right? This one started with histidine, serine, cysteine, tryptophan. This one's going to start with proline, glutamic acid, phenylalanine. It has relatively more hydrophobic amino acids. So when we put them together, boom. Okay, and it looks different. All right. Last one here for Unit 1, the chemistry of life, right? We are looking at nucleic acids, the structural differences between DNA and RNA. Okay, DNA and RNA, what is their function? The function of DNA is to store hereditary information, right? You got your DNA from your parents, and it's going to code for who you are. RNA is going to serve as also as hereditary information. It's going to help transfer the information in DNA into proteins. right? And proteins are what do the work in our body. RNA can leave the nucleus, whereas DNA can't. So how does the structure of it lead to its function? Well, the structure of it is it has a very strong backbone, a very strong sugar phosphate backbone that are covalently bonded. And then in the inside, it's going to have these nitrogenous bases, A's, T's, G's, and C's. And these nitrogenous bases are held together by weaker hydrogen bonds. And these hydrogen bonds can be broken by enzymes such as RNA polymerase that are going to transcribe genes, or DNA polymerase that are going to copy it. And so the information on the inside can be accessed, and then it can be made into proteins. There's also some inherent mutability in DNA, right? That when it's being replicated or copied, things might get changed and they might get altered. Usually mutations are harmful, but sometimes they're good. And so if we started out in evolution as the Nucleo, the last universal common ancestor, mutations provided the raw material for evolution to act upon. They provided different alleles. Let's look at a nucleotide. One nucleotide is like the Lego, it's the monomer. It's going to be composed of a phosphate group, a sugar, and a nitrogenous base. All right, and those bases can be A's, T's, C's, or G's. That'd be adenine, thymine, cytosine, or guanine for DNA. And in RNA, there's no thymine. It's replaced with U, so it'd be adenine, uracil, cytosine, or guanine for RNA. The A's and G's are called purines, and they're going to have a double ring structure. I remember this by AG stands for silver, and it's pure. And so you can also then remember that the pyrimidines are going to be smaller, and they are cut. They're cut like a pyramid. Get it? I thought that was pretty good. The big part here is that a pyrimidine must match with a purine in order for DNA to have the right size and shape. And the ones that match are A's go to T's and C's go to G's. So look, a pyrimidine is going to match with a purine. Pyrimidine, purine. Same thing with, and then in RNA, the T is replaced with a U. Let's see. We can see it in action here. I think I have one. A's and T's are going to have 2 hydrogen bonds formed between them, and C's and G's will have 3 hydrogen bonds formed between them. All right, what are some differences between DNA and RNA? Well, DNA is double-stranded. It's going to stay inside the nucleus in a eukaryotic cell. RNA is single-stranded. It can leave the nucleus in a eukaryotic cell and will go out into the cytoplasm, and it will eventually be degraded by enzymes. It'll be broken down. They have a different sugar. Look at the sugar and see if you can figure out a difference between them. Well, here in deoxyribose, it has 1 less oxygen than in ribose. And so this provides a difference for them. They also use different bases. Okay, let's look at DNA. We say that DNA has directionality. It's going to have a 3' end and a 5' end, and it's going to be anti-parallel. You can look at my arms and see they're anti-parallel. This one's going this way, this one's going this way. You can think about it like a highway. So like Salt and Caboose Highway here in Muscat. What cars are going to go here and the other cars are going this way. You can only add, or DNA can only add new nucleotides to the 3' end. And the reason there is it has an OH here. And as you know from earlier in the video, the OH is primed for dehydration synthesis. You cannot add a nucleotide to the 5' end. So this gives DNA some directionality. You can only extend it this way. You can only extend it this way. Okay? Oftentimes, let's see, what do I have? We have a picture here of transcription, right, of making an mRNA. And so here you can see that the RNA is the same way. It's only added to on the 3' end. We'll get into this a lot with Unit 6. I wouldn't sweat too much about it if this is confusing you a bit. All right, that was a long video. That's it for Unit 1. I noticed that as I went through this, I didn't really go over acids or bases, so I'm going to make a quick little tutorial on that as well.